So last 2 lectures, this is what the next exam is on. So don't freak out about the last exam because you can drop it. So the final is on the 15th, 3 to 5 p.m. So make sure you get here at 3, not 3:30, or else you'll lose a half an hour. So the average for the exam, actually it was a 76. The median was actually 77. That's better than the last exam. I always shoot for around 75. So we're still doing better. I know some people are disappointed. The range was a 37 to a 103. So there were people getting hundreds and over hundreds. But there were people getting lower than that. The problem is the median was at 77, which is why so many people are disappointed. But I guess that's how it goes. Okay, here we go. So, defects in the DNA repair system are associated with a number of syndromes and also associated with a higher incidence in developing cancer. And so this is a list out of the textbook of about 1, 2, 3, 4, 5, 6, 8 or so syndromes that are associated with defects in DNA repair. Kind of makes sense that if your DNA repair machinery is not working, you're going to have trouble, right? Does that make sense to people? Because you're going to accumulate different types of DNA damage just by going through life walking in the sun. And so you need these repair systems so that you don't accumulate mutations. Okay. Things that you may have heard of before. I don't know. Well, there's one, the BRCA2. Remember, you guys have probably heard about that in terms of its association with breast, ovarian, prostate cancer. And so that affects homologous recombination. But a lot of these you may have heard of before. Okay, so if you don't repair your DNA, then you're going to get base deletion or substitution in the daughter DNA molecule after DNA replication. So, for example, if you get deamination, and so that's a kind of DNA damage, right? And then you're going to get DNA damage. So your deaminated cytosine is going to become a uracil, right? And so then upon DNA replication, you'll have one daughter's DNA molecule that's totally fine because that G wasn't messed with, so it's going to base pair with that cytosine and you're all good. But in your other molecule is going to have an A added in there because that's what's going to base pair with the U. And so you end up accumulating mutations this way. Here, if you have a depurinated A, so now you have an A base 6 site, that means a site missing a base. Then when you have DNA replication, then you're going to have one daughter molecule that's fine, because that's going to be coming from your T. And then here you're going to have, there was nothing here, right? So you're not going to add anything, so you'll end up with a deletion there. Okay? Yes?
We know who DNA, right?
DNA.
So
DNA
where did the uracil even come from? Is it
Deamination of your cytosine. So if you remove an amine group, it becomes a uracil. That's one way, that's one form of DNA damage, is deamination. So you can see that you need repair systems. Otherwise, when you have DNA replication, you're going to get accumulation of a lot of mutations and deletion. So there's this spontaneous DNA damage that just kind of happens just normally, right? You don't have to be getting X-rays to get DNA damage. You can get oxidative damage that's happening from just reactive metabolites, so byproducts of your metabolism of the foods you eat, and then different environmental chemicals. You can get hydrolytic attack, uncontrolled methylation. So these are things that are just happening. And so these repair systems help to correct the damage so that you can replicate your DNA and you don't accumulate mutations. So about 500 purines are depurinated, and about 100 cytosines are deaminated per cell every day. So there's a huge amount of DNA damage happening right now in your body. So this is showing what depurination, so that's removing a purine. And so you end up with an abasic site. And then this is deamination, like what we were just talking about, which is where you remove this amino group and you end up with a uracil, which shouldn't be in DNA, right? So just going running outside on a nice sunny day, you're going to end up with DNA damage because the UV radiation from the sun can produce covalent linkages between two pyrimidines, T's and C's, and that can form pyrimidine dimers. And so that's showing pyrimidine dimers right here. And that's me when I go running these days because I never get out to go running as much as I should. So I always look like that. I turn into a man with a long tongue, like a giraffe. I was watching Curious George this morning. No, not Curious George. Was it Cat in the Hat? I don't know. Something with my son. While I was waiting for my husband to come home. And I didn't realize that giraffes have very long tongues. Did you guys know that?
Yes.
Because they have these leaves and I don't know. Anyways, okay, so every time I talk to anyone who's an organismal biologist, they're like, you need an organismal class. So clearly. Okay, so alkylating agents are byproducts of metabolism. And what they do is they alkylate, right? So they add methyl or ethyl groups to bases. And so this is showing you can have a guanine, and then you can get addition of that methyl group. So you'll get O6-methylguanine. And then when you have DNA replication, that O6-methylguanine wants to pair with a T instead of a C, again, kind of propagating this increase in mutation. So then on a second round of mutation, the T is going to pair with an A, so you have a GC becoming an AT, changing the sequence. And so it can also cause interstrand crosslinks, so that's your two different strands of DNA are becoming covalently bound to each other, and that's kind of a problem, right, when you need to separate your double-stranded DNA. When do you need to separate your double-stranded DNA? DNA synthesis, right. So if you've got suddenly your base pairs are covalently bound to each other, that's going to cause some trouble. So one example of an alkylating agent is nitrosamines. And 1 year I went on a tirade, you may have heard it, I don't know if it was last year or the year before, because Towson University uses an excessive amount of bacon. It makes me crazy. I don't eat bacon. It makes me really crazy. You can't get anything without bacon being thrown on it. Anyways, next time you go to Brook Street, which I heard is becoming an Oban Pond. Oban Pond next year.
What's the Oban Pond?
Grab and go? Yeah, so they're basically outsourcing the food there. But anyways, yeah.
I'm just gonna say, Towson's affinity for bacon is the reason why I came.
Oh, that's why you came here.
Yeah.
You know what? I have a student who came to Towson for the food, so I totally get that. I know it's weird, right? I told her she had issues because the food's awesome.
She's hungry.
She was impressed by the food. But no, seriously though, so cured meats, including bacon, contain nitrosamines. Meats contain amines, sodium nitrite, and that's a source of nitrosating agents. And so it's added as a preservative to meats. But don't worry, you can counteract this by taking your ascorbic acid. So next time you have your bacon sandwich, have it with a glass of orange juice. Or take a vitamin C. But yeah, so this is one source of DNA damage that's coming from just your normal diet. Intercalating agents are aromatic or flat hydrophobic molecules that become inserted between the stacked bases and the double helix. And so one very common one that we use in the lab is ethidium bromide, and it's used to stain DNA. And it actually slides right in there. So you can see in this picture here, here's your ethidium bromide, and it slides right in between the bases. The pink is your ethidium bromide. So it stains the DNA quite nicely. So then you have ionizing radiation, which is what this week's paper was about. Who did their recitation paper? I've had a lot of people visiting me and emailing me about it. But what they're talking about with those double-strand breaks is double-strand breaks caused by ionizing radiation. That's why they're irradiating their cells to cause the damage, because you can't really study the DNA damage response if you have no damage. So they're inducing the damage with gamma irradiation. And so the way it works, someone asked me today how that works, and so basically you have this kind of these high energy particles that are being released by the gamma radiation, and that'll liberate an electron and break your bonds between your bases. And so you get double strand breaks that way. Okay, so now everyone's scared. Is everyone scared of going for a run while eating bacon because they might fall and break their leg and need an x-ray? I'm terrified. I'm actually scared of everything now with what happened with my kids and the E. coli. I wash my hands so much that I'm going to need to be medicated, I think. Seriously, no, I'm like out of control.
You're losing your skin. You need to
I'm out of control with the hand washing. But anyways, once you've gone through what I've been through. No, but seriously, damage is occurring all the time, right? And it seems like it's pretty detrimental. So why have we survived? Why has anyone survived? Any species survived? Right, because there are repair mechanisms. So there's all this damage. So we needed to evolve repair mechanisms. Otherwise, we wouldn't survive if our DNA was broken all over the place and stuck together and all sorts of bad things. If replication couldn't happen. So, right, so there's two reasons that we're going to be okay. One, we have our genetic information stored in double-strand DNA. So you've got a template for repair. So that's good. And two, there are many different DNA repair mechanisms. So you can go running with your BLT, but you might choke. Don't go running with the BLT. Okay, so there's two main categories of DNA repair mechanisms. And this is a question that everyone's just giving me a hard time about. And so you're always welcome, it's a little late now, but you're always welcome to listen to future lectures in advance because sometimes you get some answers. But two main mechanisms. One is reversal. So that's specific enzymes that recognize damage and reverse it. So those two examples are photolysases, and they use energy from near UV light to split pyrimidine dimers. And the other one is our methyltransferases, and they recognize methylguanine or methylated backbone and remove the methyl group. So it's just reversal. And the other one is excision followed by resynthesis. So you're actually cutting out your damage and using a polymerase to resynthesize. Yes? They remove the methyl groups from your guanines, from your methyl guanine, or from your backbone. Yep. Okay, so here's an example of reversal by methyltransferase. So ADA, or ADA, or ADA, is an alkyl transferase in E. coli. So you know from the words, it's an alkyl transferase, so it removes alkyl groups. And so it'll remove a methyl group from DNA, And it also activates its own expression, which I think is pretty cool. So it activates its own expression and expression of ALK-A, which is a glycosylase that removes methylated guanine. So you'll have your methyl groups added to your DNA, and this isn't the kind of methylation that we've been talking about with silencing or all that. This is methylated guanine. Okay? So what kind of methylation are we normally talking about with, right, cytosine with silencing? We have cytosine, methyl cytosine, right? So this is a different, this is O6 methyl guanine. So you'll have your methylated guanines, and then your eta is going to bind to and transfer a methyl group from the DNA to its own active site. And then is able to go bind and activate expression of its own gene and the related gene, a glycosylase, beta and alpha. So that's kind of cool, right? That's that kind of positive feedback, I guess. So it's a single-use enzyme because the removed methyl group is covalently bound to a cytosine. Let's try it again. Cysteine in the enzyme's active site. Oh God, who's ready for the end of the semester? I'm so ready. Okay. Nothing to do with you. It all is having to do with Shiga toxin. Yeah.
Still?
Yes, my son is still out of school.
Is that cysteine supposed to say guanine?
No, cysteine. Yep, no, it's cysteine, because what it's doing is it's taking that methyl group off the guanine and adding it to its cysteine in its active site. Okay. So now we can talk about removal of replication errors. And so one mechanism for removing, well, first of all, should we have many replication errors? No, why? What keeps our replication errors low?
The exonuclease.
Proofreading, exonuclease activity that is intrinsic to your DNA polymerase. So ideally, we don't have that many replication errors, but they can happen. And so we need a way to fix them, right? So there's a process called strand-directed mismatch repair. And so, or post-replication mismatch repair, same thing. And so what it does is it repairs a mismatched base that's missed by the exonuclease activity. So what you'll have is some kind of error in your newly made strand of DNA. And so MUT-S will recognize this effect on the DNA structure. Because when you have incorrect base pairing, the structure of the DNA is going to be affected. It's not going to be, it's going to bulge out a little bit. And so MUT-S is going to recognize it by recognizing the change in the DNA structure, and it interacts with the sliding plant. So it's also close by and able to detect changes in the DNA structure. And so the new strand is distinguished from the parental strand. How? How is the new strand of DNA distinguished from the parental strand? Methylation, right, right. New strands of, remember we learned about hemimethylated DNA is the result of DNA replication in bacteria because you get methylation of your DNA, but there's a little bit of a lag time. And so during DNA replication, your new DNA is not going to be, your new strand of DNA is not going to be methylated, but the parental one will. So you'll have this hemimethylated, and that's one way to recognize a template strand. Is there a party going on that day? I think there is a party. Everyone's so excited for the end of the semester. They're walking by and they're like, woo! So helicase 2 will then be recruited and it displaces the nicked strand. So there could be a nick. So the DNA scanning is also going to detect nicks in the new DNA strand. Where are those nicks coming from in your new DNA strand? Okazaki fragments that haven't sealed yet, right. So then exonucleases are going to degrade the DNA between the nick and the damage, right? So you've got a nick from your Okazaki fragments that aren't ligated yet, and then you've got your damage because your exonuclease activity didn't work. So then your exonuclease activity is going to, your exonucleases that are distinct from the activity of DNA polymerase is going to degrade the DNA between the mismatch and the nick. And then you get resynthesis using the template strand, the correct strand, and then it gets ligated to the DNA. And also there's another protein called MUT-H, which nicks unmethylated GATC sequence in bacteria. So that's one. So just if you're keeping track, we've talked about reversal, which was really just your photolysis and methyltransferases, and I gave you this example of this, the ADA. And so now we're going to be starting to talk more about excision and resynthesis, right? So this is excision and resynthesis, right? Because you're excising a chunk of DNA and you're resynthesizing using a template that's correct. So there's also mismatch proofreading system in humans, and it will also recognize the template strand because of those nicks, because the nicks haven't yet been sealed by DNA ligase. And defects in this system are associated with a hereditary form of colon cancer, where you get rapid accumulation of mutations. Why would you get rapid accumulation of mutations if you have a defect in mismatch repair? Yeah.
But if it's one mistake, you can't really fix that. It would like multiply, and then it would just goes on and on and on.
So yeah, well, so if you can't repair when DNA polymerase adds a wrong base, then that's gonna be passed on to daughter cells. And if that happens fairly frequently, well, then you're going to accumulate a lot of mutations. Did I see a hand going up over here? No. Okay. So mismatch repair also corrects insertions and deletions that come from what's called slipped strand replication. And so what could happen sometimes during DNA replication is you can get a secondary structure that can form in the DNA, in the single strand DNA, that will result in insertions or deletions. So for example, you might get this kind of base pairing between repetitive sequences, and then when you replicate again, it's going to delete that whole sub-loop structure. Yeah. Okay, so let's talk about excision repair. So there's two types. There's base excision repair and there's nucleotide excision repair, which again, you guys some people got upset about this question too on the quiz, so I am looking into it. Hold your comments. Sorry. So with base excision repair, it's just removal of one base. And so this is often, so in this case, for example, if you have deamination and you're going to remove your uracil that shouldn't be in DNA. So that's done by UDG, uracil DNA glycosylase, yes.
So if you have thymine dimerization, what would you use?
Apparently, NER.
Okay.
Yeah. But so it all depends on how much needs to be removed. And so if just one base can be removed, you're going to have your BER, base excision repair, and you remove your one base. And so what happens is you get removal of your base, so you have an abasic site, and then another endonuclease is going to remove your backbone, and then the DNA polymerase is going to come in and use your template to repair it. Okay? And then with nucleotide excision repair, here's where you might have your pyrimidine dimers. So here you can't just remove one base, you need to remove multiple bases. So you'll use this repair mechanism where you'll have a nuclease that will nick 2 places on the strand, and then that whole stretch of bases, maybe 10 bases or so, will be removed. And then your DNA polymerase is going to come in and use a template to repair it. Any questions? So damaged DNA bases and A basic sites are both repaired by BER. So anytime you can get away with removing one base, right? And so you can generate an A basic site just by breaking a glycosidic bond between your base and your sugar phosphate backbone, and so that'll be removed, and then the gap will be repaired by DNA polymerase. And so then you can also have a damaged base that will be removed by DNA glycosylase, and that creates a damaged site, a basic site, and that'll also be repaired by the same mechanism.
Instead of like breaking like acidic bond called depurination.
No, because that's just so depurination. That's just moving your impurities, but actually removing your whole phosphate backbone is that's breaking your glycosidic bond. Right? Am I right? No. I'm sorry. We're making your glycosidic bond is between your backbone and your base. So that creates an A basic site. Yeah. Okay. So damaged bases are removed by DNA glycosylase, which creates your A basic site. So here's for example, here's a G that's damaged. It's got some kind of methyl group, let's say, added to it. And it's going to flip it out and remove that base. And so now you have an A basic site. A base there. So that's base excision repair. Okay, because then you have your polymerase can come in, use your template, use your correct template strand as a template to repair it. So with nucleotide excision repair, you're going to get removal of a short stretch of DNA that contains bulky lesions, such as thymine dimers or something like that. And so these bulky lesions are usually produced by UV radiation or chemicals, and they're going to create a distortion in your double helix, and that's how it's recognized. So you can see here, this is showing a bulky DNA lesion. And what's going to happen is you're going to get removal of 10 to 30 base pairs of DNA. And so in the mechanism, you have UVR proteins, and they're UVRA and B, which are going to scan for the damage. Then you have UVRB is a helicase that opens up the DNA. UVRC is going to nick on either side. And then UVRD and RNA Pol 1 are going to release, DNA Pol 1 are going to release the oligonucleotide, and then you get repair by resynthesis. So in different organisms, you're going to have different proteins that are responsible for carrying out nucleotide excision repair. You're going to have different names. So all those UVR proteins are all in E. coli. In yeast, it's all called the RAD proteins. And then in humans, they tend to be called XP something. And that has to do with its association with the disease called xeroderma pigmentosum. Yep. Same functions though, which is cut out a bunch of DNA to some extent. Okay. So now we're going to move on to double strand breaks. So we went through just kind of reversal mechanisms, and then we talked about We talked about removal of errors from replication. We talked about base excision repair and nucleotide excision repair. And so now we need to deal with double strand breaks. And these are slightly more involved, kind of. There's two different mechanisms for repairing double strand breaks. And they're both excision and resynthesis. They both fall under that category. Because you have got non-homologous end joining and you've got homologous recombination. In both cases, you need to excise your ends and then resynthesize to fill in. The only difference between non-homologous end joining and homologous recombination is whether it's template directed or not. Homologous recombination is using it's using information that's coming from the sister chromatid. And so this is kind of cell-cycle dependent, right? Because you always have sister chromatids sitting around. Now, when do we have sister chromatids sitting around? Mitosis. So basically from end of S phase until chromosome segregation, I guess. Right. So you're kind of limited as to when you can use your homologous recombination because you need that sister chromatid as your template. Non-homologous end joining is not template directed, so you actually are going to kind of accumulate mutations this way. So remember, double strand breaks are caused by ionizing radiation, and also you can have them caused by some replication errors and oxidizing agents and metabolites, kind of all. But usually when I think double strand breaks, I mostly think of ionizing radiation. Okay, so I think of non-homologous end joining as the quick and dirty. Basically, all that's happening is you're having trimming of your ends, and then some nucleotides are added in. And so you don't need a template, but you also don't maintain your sequence. So in this figure showing there's these Ku proteins that are going to recognize and bind to the broken ends. And then you're going to end up having some other proteins involved also. And then you get processing of the ends, and then you get repair synthesis. So basically, a little bit of repair synthesis and then ligation. So you actually lose some sequence. This is fairly common in mammalian somatic cells. So by the age of 70, the typical somatic cell contains more than 2,000 non-homologous end joining scars. And so you can detect them by the type of the loss of sequence, basically. And then this is also a source of chromosome translocations. Okay, so then we've got our much more accurate method of double-strand break repair, which is homologous recombination. And you guys learned about this probably in genetics, right? Because when else do we have homologous recombination? Crossing over? Yep. Right. But this is also used for DNA repair. And so it's accurate because you're using your template from your sister chromatid. And so it's occurring after replication. It's often used to repair broken replication forks. And it's also used in other gene rearrangements. And so basically what you have is you've got this trimming of your 5' ends, and then you get this strand invasion where one strand enters the sister chromatid DNA. And so for a short period of time, you have this kind of 4-strand DNA molecule. And so then you're going to have, so it's using that. So basically you're going to have base pairing of newly synthesized DNA with the, pairing of the DNA with the sister chromatid DNA, and then you're going to get resynthesis, and then you're going to get resolution, so it has to come back out. You can't always remain this 4-stranded DNA, and then you'll get ligation. and then you're good to go and you have accurate repair. Okay, so the basics of this is you've got your break, you have your trimming, so you have the 5' ends being trimmed, and then that 3' end is going to invade your sister chromatid and base pair with that homologous sequence, and that's called a branch point, branch migration. And then the DNA is repaired using the template from the sister chromatid. So here's your 4-stranded DNA, which is also called a holiday junction. And then you get resolution of your holiday junction. So you come back out and you go back into 2 different chromosomes, double-strand DNA, and then you're going to get ligation. And then you're repaired. Yes?
So those are 2 different, like totally different chromosomes or strands interacting, like there's one pair interacting with a whole different other pair?
Yeah, it's interacting with the sister chromatid.
It's not the whole different thing, it's a sister chromosomes.
It's a different DNA molecule because you have a double-strand break, so you have no template, right? Usually, so with NER and BER, you only have one strand being affected, so it's easy to use the other strand as your template for repair.
So how does it know which exact area of that other sister chromosome to use in order to repair the part that's missing?
Homologous, it looks for homologous region. So remember, 2 sister chromatids have the same genes, right?
Yeah.
So they were just replicated from each other. So you've got the same genes, same sequences. It's often involved with replication fork. So ideally, if you have a double-strand break, everything's right there. And so then you can have strand invasion into the same sequence.
So is it also between S phase and R?
Huh?
Is it also between S phase, the end of S
So
phase
basically, it's happening after replication or during replication. I don't know the exact. I think if we went deeper into the exact mechanism, like this is a very kind of superficial mechanism. There are probably other proteins involved that must have something to do with how the different components interact with the replication fork, or the DNA replication machinery, stuff like that. But I don't know exactly. Any other questions? Yes.
So, if we're talking about single strand breaks, we look at nucleotide excision repair and base excision repair, and if we're talking about double strand breaks, it's the non-homologous end joining and homologous combination.
Right, so double strand breaks, always non-homologous end joining and homologous recombination. I would say single strand breaks, I would say damage that affects one strand. Because it's not a break, really. Actually, the break is being made by an enzyme that's being recruited to remove the base. Yeah, but yeah, so that's a good way to start organizing yourself for the test that's coming in 5 seconds. Yeah. Okay. So Rec A, or RAD51 in eukaryotes, is a DNA binding protein that catalyzes strand invasion. So you guys have heard of Rec A before, right?
It should be the paper.
In DNA replication, single strand binding protein. So Rec A intertwines with single strand DNA and the double strand, and it intertwines it with the double strand template. So it actually, the single strand DNA is then going to get in there and search for the homologous sequence. So this kind of addresses what you are asking. The single strand DNA is going to base pair. So how is it, like, it's not going to want to bind very tightly if it's not base pairing, right? So it's going to base pair with this strand from the double strand DNA of the sister chromatid. And so that's Rec A and RAD51. Okay, so sometimes you have a situation where you can't get repair for whatever reason. And error-prone DNA replication is better than no replication at all. So we have translesion DNA synthesis. And so basically what it does is it bypasses DNA damage that stalls the replication for. So normally you might have, so here's your sequence and you're gonna have some DNA damage which causes this big X here. And so during DNA replication, you're gonna have replication and it's gonna come up to the point of damage and stall. And so then translesion polymerase, so a different DNA polymerase, is going to be recruited and basically do non-templated DNA synthesis just to get past that damage. And then you're going to have, then that translesion polymerase is going to leave, and your DNA polymerase is going to return, your normal replicative DNA polymerase. So this is really more, not so much a repair mechanism, because is repair happening here? No, so this is really damage tolerance. And so we have to put up with a certain amount of damage, because if we don't replicate, do we survive? No, right? So it's low fidelity, obviously, because it's non-templated. Poor processivity. What does processivity mean? How well it stays on. Do you want translesion polymerase to stay on very well for a long time? Absolutely not. You want it to come on, fix that one area of damage, and leave. And then there's no 3' to 5' exonuclease activity. Also kind of makes sense because what you're doing is tolerating damage. It's got a much larger active site, so it can tolerate kind of bulky lesions. Its error rate is 10 to the negative 2, 10 to the negative 4, compared to 10 to the negative 5, negative 6. So it's much more error prone, but who really cares? Because it's non-processive and it's tolerating damage. Right? So if you look at the structure of translesion polymerase, if you compare the active site of your normal replicative DNA polymerases with your with that of the translesion polymerase, it has a much larger active site, and so that kind of makes sense because you're going to want to tolerate bulky lesions. Yes.
So what's activating these translesion polymerases? Is it that REC-A protein?
No, REC-A is allowing for strand invasion in homologous recombination. That's what REC-A is doing. What's happening here is it's actually translesion polymerase is being recruited specifically as part of the DNA damage response to the sliding clamp. So, because remember, when it gets recruited is when you have a stalled replication fork. And so, I don't have the exact mechanism of regulation on here, but you have to guess, actually this might, right. So, and you guess this has to be tightly regulated because you always want your translesion polymerase coming and binding to your clamp. No, because you don't want it to start adding in random bases to your DNA. So I don't have the exact mechanism here, but if you were a scientist, not if, since you are a scientist, if you were going into a lab for your PhD, MD, whatever, if you're going to go do your MD, you'll probably want to get research experience too, so we're going to throw you all in here together. And you go and you work with someone who's working on translesion polymerase, and they say, okay, we know it's recruited to the clamp, but we don't know how. And you want to you decide you're going to kind of sit with your lab mates and brainstorm what kind of mechanisms might be responsible for recruitment of translesion polymerase to stall replication fork. So what kind of things would you think? You know it's going to be recruited to the clamp, right? But you know that's highly regulated. What kind of things would you think about in terms of some kind of change that might happen to regulate whether or not the polymerase is recruited to clamp? I just heard phosphorylation.
Yeah.
Great, so you guys are on it. Exactly. I don't know that it's phosphorylation, but some kind of post-translational modification that's going to suddenly create a binding site for translesion polymerase. I don't know that this is right. We can look it up and see. But actually, that's what we'll do since you're all angry about the quiz. And because I'm so nice and you're all desperate for extra points, let's make this a hopefully last ever Stump the Teacher question, because I stumped myself. It's not so much that anyone stumped me, actually. It's just going to be a challenge question, which is what's the mechanism behind recruitment of translesion polymerase to the sliding clamp at stalled replication forks? I need an exact mechanism or what is known, or as much as is known.
I kind of lost you after mechanism.
Come on, you should be ready for it.
What's that?
Okay. Mechanism for recruitment, behind the mechanism of recruitment of translesion polymerase to the sliding clamp when it's needed, such as at stalled replication forks. And so just to make you guys happy, because that's all I live for, is making my students happy. It's like my children. No, just kidding. It'll be worth 2 points.
Oh,
Yeah.
Thank you.
Well, that's really because everyone's pissed off about the quiz that I wrote way too quickly. So tomorrow, 5 p.m., don't complain about anything ever again. And you know what? The people who aren't here don't get it. So there you go. I mean, they'll listen to this, but probably not until it's over. Are you going to say ha-ha? Is it? Uh-oh, you guys are being mocked. Yeah.
This is business. It's about to
Yeah. Let's get through this and then we'll talk towards the end, okay? Okay. So I just went through a bunch of different types of damage and the main mechanisms for repair. And so now we're talking about the DNA damage response. And this is what the paper was about, right? If you have damage, you need to somehow signal that there's damage and recruit the repair proteins to the appropriate location so that repair can happen. And that's your DNA damage response, right? And so your DNA damage response is going to be induced by excessive damage or damage that interferes with DNA replication. And the purpose, recruit repair proteins, amplify the response, and then halt the cell cycle for repair. Right, so we talked already a little bit how damage affects DNA replication, right? Because you'll get stalled or collapsed replication forks. And so that's one sign of DNA damage. So here you might have some kind of lesion. A lesion is when you have something added on to a base that causes some kind of wrong conformation of your DNA. That's usually referred to as a lesion, so it's a DNA damage lesion. And so that when you have replication, you're going to get the fork is going to stall there because that lesion's in the way and it's interfering with DNA replication. And so you're going to get an excessive amount of single-strand DNA there for a long period of time, longer period of time. Or what if you have a nick in a single strand, and then your replication fork's going to approach that nick, and what's going to happen? Your replication fork's going to collapse. So those are two situations that are going to signal the DNA damage response. So lots of single-strand DNA, collapsed replication forks, DNA ends that are just naked and hanging out there. So in multi, yep.
Sorry. So how is that connected to RNA polymerase?
How is it connected to RNA polymerase? Oh, because of that paper I was telling you about that another student sent. Well, so let's say you have, okay, so there was a paper, well, and also, so you can also have the DNA damage or damage response signal activated by stalled replication forks due to RNA polymerase. And that, not replication forks, inhibition of RNA polymerase, not really a replication fork. But if RNA polymerase is going through the DNA, because remember, you have your double strands separating and your RNA polymerase is going to go through. If you have something that's preventing separation of your double strand, your DNA, then that's going to signal. And that's going to signal the DNA damage response as well. But that wasn't what I was trying to do on that quiz. Yes?
So the single strand of DNA results in recruitment of the DNA damage response gene. Is it possible to get like the single strand DNA into the secondary structure just like you get to RNA?
Is it possible that you're going to get formation of a secondary structure in single strand DNA, just like RNA? Well, you've got all these single strand binding proteins that are supposed to be preventing those things, but you can get some, as I saw, like there's this slip strand synthesis where you will get a small region of secondary structure that will result in like deletion of a whole bunch of bases. What were you going to say?
No, just something. Where would you place microRNA and siRNA into that? Would you say that they play any role in?
Do microRNAs play any role in
No,
the
because you're saying about single-strand DNA and microRNA at some point combined, right?
No, microRNAs bind to RNA,
RNA.
mRNA. Yep. But that's good. You should always be trying to connect the different things that we're learning. Okay, so in multicellular organisms, if you don't repair your DNA damage, then apoptosis is triggered because, well, it depends within reason. But if there is no repair, you would like to trigger apoptosis because accumulation of a lot of mutations is not good either. So, okay. So you know what? I feel like no, we'll go on for a little bit. Okay. So in bacteria, there's the SOS response. Okay, so this is the bacterial response to DNA damage. Quite different from eukaryotic cells, but basically what happens is normally you've got this REC-A. This was this single-strand DNA binding protein, right? And am I right? REC-A. Isn't that what we just talked about?
Yeah.
Yeah. And normally it's inactive. You have your normal replication, and this LEX-A is a repressor that's repressing expression of your SOS genes because there's nothing wrong, and your SOS genes are part of the DNA damage response. Okay? But if you have some kind of damage that results in a stalled replication fork, you're going to have an excess of single-strand DNA, and it's going to get bound by REC-A, and that REC-A is then going to bind to and inactivate LEX-A. So what kind of protein? Oh, I have it written there. LEX-A is a repressor. I was going to ask you what it was, but it says it right there. LEX-A is a repressor, but when you inactivate it by cleaving it, then you're going to get gene expression. And you're going to get expression of a bunch of genes, more than 40 genes, including these UBR proteins, translesion polymerase, for example. And then you're going to get transcription of more REC-A, and then what you're going to have is, once you have repair, then REC-A is going to be inhibited again. So it turns on gene expression, it produces its own inhibitor, you get repair, and then you can inhibit REC-A, and then LEX-A is able to bind again to the operator and repress gene expression. Yes?
So is Lexa always bound unless there's DNA or DNA?
You tell me. So if LEX-A is a repressor, do you want it repressing expression of DNA repair genes if there's no DNA damage? Yeah, so you should, right? Because LEX-A, so if LEX-A is not bound, then you get expression of all these genes to repair the DNA. And so that's how that's related. So when you've got your REC-A bound to that single strand DNA, it's gonna basically inactivate the repressor. So you can connect this to what we did in the last exam, right? Where we talked about repressors and things that regulate whether it can bind or not. Here's just the mechanism for regulating whether the repressor can bind or not. And really what's happening is you're inactivating it in a way so it can't bind. And the thing that's inactivating it is your single strand DNA binding proteins. And then it turns itself off. Okay. Yes.
Where do you have the right, the question, where else do you have?
Well, I answered it for you by accident. Where else have we seen REC-A?
Wasn't it double-stranded DNA damage?
So so we saw it today when we were just right there in homologous recombination. It's a single strand DNA binding protein. So we saw it here in strand evasion, and then we also saw it in DNA replication because it's going to bind to single strand DNA during DNA replication. The difference is you shouldn't have a long stretch of REC-A hanging out for a long time in DNA replication because your replication fork is moving. But when you have a stalled replication fork, then you kind of generate this long stretch that's just sitting there and can be detected. Okay. So once DNA damage is repaired, REC-A can bind to DIN1, which is a DNA mimic. So basically, it's a protein that has a structure that's similar to DNA and shuts off the DNA damage response. So let's go back and look at that. REC-A likes to bind to single-strand DNA, right? DIN1 is induced by the SOS response, so that's negative regulation. And so then DIN1 is going to bind to REC-A and take it away so it can no longer activate that SOS response. So that allows shut off. And so then you have LEX-A that's able to bind and repress again. Okay? So you have this negatively charged alpha helix in DIN1, and it actually looks like DNA, single-strand DNA, which is how REC-A binds to it. Okay? Any questions? Okay. So the general DNA damage response, again, relate this to the paper for this week. It's really just about recruiting repair proteins to DNA damage, the site of DNA damage. This is in eukaryotic cells that we're talking about now, right? You need to sense, there are things that need to happen, right? You need to sense either single-strand DNA or double-strand breaks, so that's your damage. And then you need to recruit kinases that are going to help signal the presence of the damage. And then those kinases are going to phosphorylate and activate other things. And then that phosphorylation creates binding sites for recruitment of repair proteins. And then you recruit checkpoint proteins. Checkpoint. That are going to halt the cell cycle. You know, when we studied cell cycle, we're talking about checkpoints, we're talking about DNA damage. I'm just saying. Bring it all together. So that allows for recruitment of repair proteins and inhibition of cell cycle progression, because do you want to allow the cell cycle to progress when you have a ton of DNA damage? No. No. Okay. Okay, so I think we'll stop here for today, but not for any reason other than there was business that was being raised in the back, and just to see if there's anything else that needs to be spoken about. Wait 1 second while I shut this off.